For my Lab 6 video presentation, I will be focusing on Lab 5, Bacterial Transformation. I'd first like to go through some background information derived from the lectures to lay out the context. What are bacteria? Bacteria are classified as prokaryotes alongside archaea and cyanobacteria, which is blue-green algae. Prokaryotes are unicellular organisms, and their cells are comparatively smaller than those of eukaryotes. They possess no nucleus and lack membrane-bound organelles. All prokaryotic cells are encased by a stiff cell wall due to the presence of peptidoglycan. The cell wall is responsible for maintaining the cell's shape, protecting the cell's interior, and preventing the cell from bursting when there is an increased uptake of water via osmosis. Moving on, what is a plasmid? A plasmid is a small circular piece of DNA which is composed of a couple thousand bases at most. It can replicate independently from the host chromosomal DNA and has an origin of replication which makes it a good cloning vector. When found in nature, such as in microbes, they contain genes that provide a competitive advantage, such as antibiotic resistance, thereby increasing its chances of survival and reproduction by binary fission. Plasmids are small, stable, and easy to manipulate. All plasmids contain an origin of replication, which instructs for the initial site of replication. They are designed to introduce genes of interest through electroporation. Restriction enzymes acting as a pair of molecular scissors recognize these sites located in multiple cloning sites and cut out the present gene to insert foreign DNA. A different gene is inserted into the site and expressed only when the promoter site, which is located upstream of the inserted gene on the plasmid, acts as a green light to allow gene transcription. Foreign DNA and plasmid vectors are both cut with the same restriction enzyme and are mixed together. However, not all molecules will join to form recombinants. Some vector molecules will reanneal without incorporating the foreign DNA. In this case, leads to no ampicillin resistance, resulting in the death of the host cell. I will now provide a quick overview of the experimental procedures carried out to practice this lab. The purpose of this lab is to learn how to produce transformants by plasmid acquirement and use the LAC-Z gene as a selectable marker. I performed cell transformation using GH5A competent cells and PUC19 plasmid. Our goal is to produce 4 different cell plates on the LB medium to understand bacterial growth and transformation. First, label the 4 plates accordingly. Next, thaw chemically competent cells on ice before pipetting them into the plus and negative labeled plasmid tubes. It is important that we make sure to only add the PUC19 plasmid solution to the positive plasmid tube. Incubate both positive and negative plasmid tubes on a bed of ice for 20 minutes. Afterwards, heat shock the 2 tubes on a float in a hot water bath set to 42 degrees Celsius for 45 seconds. Pipette the nutrient LB medium into both tubes and mix thoroughly by tapping your fingers against the tubes before incubating them for 20 minutes in an incubator machine set at 37 degrees Celsius and 200 RPM. Afterwards, centrifuge the incubated tubes and remove a portion of the supernatant. Add egg scale to both tubes and resuspend the pellet into the liquid by gently pipetting up and down. Transfer 100 microliters each from the positive plasmid tube to the 2 positive plasmid plates, and 100 microliters each from the negative plasmid tube to the remaining 2 negative plasmid plates. Use sterile glass beads to equally disperse the solution by shaking in a rattling motion. Finally, stack and tape the 4 plates together before incubating them for a day at 37 degrees Celsius. In this segment, I would like to explain about the discussion portion of the lab. Antibiotic-resistant genes are often used as selectable markers to identify whether a cell is a transformant. We can deduce that only transformants, which are cells that have acquired the plasmids, are able to colonize in a medium containing antibiotics. The PUC19 plasmid contains 2 genes, the AMPR gene and the LAC-Z gene. The AMPR gene encodes for beta-lactamase, which is an enzyme that breaks down the antibiotic ampicillin before it inhibits cell wall synthesis. The lactazine gene, however, is only expressed in the presence of galactose. However, in this experiment, we will be using IPGG to induce lactazine gene expression to produce the BIOL enzyme. BIOL will hydrolyze X-gal, which is a chromogenic substrate, resulting in the production of an insoluble pigment that will stain the bacterial colonies blue. If the colonies are white, this means that new DNA has been incorporated as the BIOL enzyme is not produced due to insertional inactivation inactivating the lactazine gene, which is also known as the second marker gene. Let's assume that the opposite scenario has occurred. If the colonies are blue, this means that there is no new DNA incorporated and BIOL enzyme is produced, resulting in the hydrolysis of X-gal, staining the bacterial colonies with the insoluble blue pigment. All in all transformants should be able to survive and reproduce by binary fission in the presence of epsilon. Additionally, they will be blue in color in the presence of IPGG and X-gal. We can apply these concepts to real life. In terms of medicine, we can produce proteins that dissolve the blood clots formed in heart therapy. Additionally, we can also produce human growth hormones that treat stunted growth. For agriculture, we can insert pest resistance genes into plants to increase the net yield in harvesting seasons. Genes can also be used to alter bacteria responsible for cleaning up toxic waste polluting the oceans. We are finally on the thank you segment. After this, you will all be shooked by my overwhelming gratitude. So let's get started. Obviously, we have to start with the one and only Professor Ju-Hyun Lee, who has enlightened the whole class with an incredible amount of knowledge in just 7 weeks. Thank you for providing me with an opportunity to apply my knowledge rather than being asked to regurgitate it. I believe I have learned the vital life skills that will definitely help me in the next 4 undergraduate years at DKU. Thank you so much for your prompt replies, helpful feedback, and extra credit opportunities. Thank you so much. I have to thank my best friend at DKU, Faith. Thank you so much for being my rock, and I'm extremely grateful for your constant support and motivation. I have to also thank my TPS Group 3 teammates, Stella, Yino, and Mushi, for their fantastic work, attitude, open-mindedness, and vocality during our biological discussions. I have very much enjoyed our conversations after we had finished our well-thought-out discussions, and I would like to thank you for being such amazing people. I hope to meet you guys soon, definitely before my senior year. Next, it would not be a thank you video without Kathy. Our weekly study sessions have helped me stay on top of my work, so I dedicate my whole final grade to you. Thank you for always answering my questions during recitation when my internet dies on me as always, and teaching me how to use Excel. I have to also thank my weekly study group from ARC, specifically Katrina, who has kept me company for the first 5 minutes before recitation starts. Finally, thank you, Lynn, for texting me the Zoom code on the first day of class and being a very good friend. Thank you. Thank you so much, Professor, for all that you do for the entire BIOL110 class. Before I end this video, I would like to apologize to everyone watching this video whom I have unintentionally deafened. Thank you so much for your patience, and I wish you all the best of luck in life.